Okay, thank you, Dr. Chen. And it's a big challenge to talking about the assembled genome after Dr. Ginny. So today, I'm going to talk about the reference genome assemblies built from the PacBio HiFi non-reads and proximity negation and optical mapping technologies for the Inbred strands, important as a module.
Show my extreme competence here. I go over here to the three little dots, and I go here, and I click.
Okay. Okay, let's go ahead. Uh, yeah, about it's really important as a module of the complex disease for the rats. My name is Kenny. I came from the University of Kentucky and based on the Global Equine Research Center. Um, oh. Okay, yeah. Why I do this research? I think it's an easy question to answer because as we know, the complete and accurate genome assemblies is from the basis of the most downstream genomic analysis and critical importance. So we can get several benefits from the enhanced accuracy of genomes. For example, we can able to study the complex genomes regions, also for the complex repeat sequence, and discover some novel gene or regulatory elements. As well, we can advance in the use of the RAS as a module. So another question is about why we use the Inbred. I think this is another easy question because we can get so many benefits from the inbred rats. For the highly inbred rats, they have no heterozygotes in the genomes, so they can cause no sequencing error or can be easy to assemble for the complex regions. Also, we can get a higher quality of the assembled genome. Before I talk about my details of the pipeline, I want to introduce two technologies. One is called PacBio HiFi long reads. It's different between the next generation sequencing technology. They make a cycle for long reads and sequencing the reads again and again and again, and then improve reads by themselves. So they increase the accuracy over 99.9% for them. So it's the Q value is over 30. And another point is the average of the length is going to be over 10 kb. Another important technologies we used is called proximity negations, HiC technology. HiC technology is a chromosome conformation capture technology. So they cross-link the DNA and the ligation of DNA to the PCR, then sequence the fragment. So all this is the pipeline I use for the new brown Norway rats compared with the brown Norway 7.2. Okay, we have the Illumina next generation reads and the PacBio continuous known reads and the Arima high series and the PacBio HiFi known reads and the bio-nano optical genome mapping technologies. So the first step, we use the high cannon with the PacBio HiFi known reads to assemble the contigs. After that, we use the bio-nano OGM to do the super scaffold. Since we get super scaffold, we polish the scaffolds. So we use the pylon with the Illumina data and use the inspector with the HiFi data. Since we get to polish the scaffolds, we use the CellSet2, these tools to scaffold our polished scaffolds. We get the HiC scaffolds. And for the next step, we're gonna use the Cannon to assemble our CLI reads. It's the continuous known reads, also from PacBio. And then we use this reads with the rectang tools to patch our scaffolds in the fellow step, we can use the rapid creation, these tools to build the fellow scaffolds. Okay, before I talking about the more detailed step by step, I can tell you some some terms about the genome assembly. First one is M50 values. Think about if we get so many contigs and we order the contigs together and by sort by the longest one to the shortest one. So can you use mouse? Yeah, okay. For example, it's the total length is the 100. It's in the middle point. It's this context. So we call this one is called the length of this one we call the M50 value, which means if the M50 value is higher, the assemble can be better. This one is settled in here. So the L50 values means the M50 the context how the number of the M50 context setting in there. So it's the 1, 2, 3, 4. So we call the L50 value is 4. We still have some other metrics to evaluate the assembles. For example, the total assembly length, we're gonna know the how long of our new assembled genome. It's error risk, it's about the accuracy, and it's the completeness of the gene or genome, or the map risk. Okay, the first step with the HiCanon and with the HiC data. HiCanon is do the 3 modules. It's create and train and assemble. Here is the results from the HiCanon. So we know here's the M50 numbers values is over 41 megabase, and its L50 is 22, means the over 21 context is bigger than the 41 megabases in our first step of the assemble. Okay, the next step, we polish our super scaffolds. We use the 2 tools, the inspector with the HiFi data, with the HiFi data again, because HiFi data is a non-read data. The non-read data gonna be help us to easily to detect the structure errors. And for the panel, we using with the next generation data. It's high accuracy, so it's easy to detect the small errors and some small gaps. Here is the output from the inspect polish. So we detected the 12 structure errors, and after polish, okay, this is for the Brownlow, a new Brownlow had polish from our process. So after that, they fix the 11 of them. And they also all can detect some small errors, and they fix most of them. And we use the Illumina short data with Python. So we know the sequence data from the rat. All of the rat, the reads cannot be homozygously against with themselves. So that's the reason if we detect some sites with the homozygous variants, so it's the error in there. So that is Python did. Python can be fix some errors from the Illumina data. Before the Python, we detected the 60,000 homozygous variants. After that is 7,000 homozygous variants. The next step is scaffold. Scaffold use the HiC data. It's the HiC data is the they capture the chromosome domain. So the data from the same domain, which means it's easy to detect the fragments from the same place. So they can be help to scaffold the context. It's M50 increase from the 41 megabase increase to the 121 megabase. It's L50 decrease from the 22 to the line. Okay, after the CELSA, we use the Canoon to scaffold the CR data. we know the CLI data is length over than 50 kilobase. So that is easy to help us to feed some gaps with the rectangle. To is some gaps we can first step with feed by n and use the CLI data compare with the the context. They're gonna fix patch and merge for the modules. Next step, we use the replications. This is the this picture is generated by pretext view. So before we get some noise from here, okay, here is the generated from the HiFi data. So from the angles, it should be, this is the same scaffolds from the begin to end. So should be arranging the angles. So this is noise and show this should be cooked and should be relocated in somewhere. So this is before and this is after. We're gonna pick our examples for the first. It's a small scaffolds in here, so we find the noise in there. So which means this small fragment should be put in there. So from the bottom of the left, it shows that this scaffold should be put in the end of the scaffolds. If they're in the top, so we should put it in the beginning of the scaffolds. If we in the right side, it shows as this is the wrong orientation. We should opposite the orientation and put the relocate the scaffolds. Okay, here is the brief new results. The new brown Norway rat with the brown Norway 7.2. We totally added 260 megabase for our new brown Norway, and it's kind of like 2.8.2%. I'm sorry, in the new genome, and the error rate is very, very low. It's just 1.3 errors per 10 megabase. It's very, very low errors, and it's completeness is the 99.7%. Okay, we keep going, do some rat genome assemble, and we have some changes in the assembly pipeline. We know the new technologies exist, not new technology. Okay, it's another technology is about the Oxford Bionano or ultra known data. So we're going to not use the CR data again anymore. And if we in the future we fix the ultra known data, we're gonna use vehicle and the bandage to assemble, to do the T2T assemble. And for the first step, we compare the HiFi with HiCanon. I want to say the HiFi is better than HiCanon because it's based on the strengths, different strengths. It's very highly depends. So, or you can compare the two results. Also, another tool is called YAS, and we also can use the YAS to do the HiFi scaffold. I want to point here because we put the panel and the inspector means the polish process before the BioNano OGM, because when you get longer scaffolds, and you can take more computer times to do the polish process. Okay, I really want to thank you, my supervisor, Dr. Ted Capfresh, sitting there, and colleagues, Dr. Peter Dawes, and Dr. Melissa Smith, and Elizabeth Hodgson. Yeah, its funding came from the NIH. And thank you for your attention.